All right, so we are here to talk about the first three learning targets, which were I can explain why some molecules can cross the cell membrane while others cannot. And then I can compare passive and active transport, and I can contrast passive and active transport. All right, so here's a cell membrane, and if you'll remember, it's made up of these phospholipids. So this is one phospholipid. And the phospholipids have two parts. They have a head, and that is the polar or hydrophilic head. And then they have tails that are hydrophobic because they are nonpolar. And because water is polar and they are nonpolar, they are scared of water. Okay? So when we talk about what can cross the membrane, small things can cross pretty easily, especially if they are nonpolar. Because if they are nonpolar and these tails are nonpolar, then there's not going to be any complaints. Now, I know the heads are polar, but the heads can kind of look the other way and let these tiny little nonpolars. Now, if it was something large, some big molecule, it's not going to get through because it would bust a hole in the membrane. So it can't come through anyway. So large is a no-go. And polar, whether it is large or small, both of those are no-goes. Okay, they're going to have to go through some kind of protein. If it is small and nonpolar, something like oxygen or CO2, those can move right straight across. And then, like I said, sometimes some polar small things can sneak across. Sometimes some water molecules can get across. But for the most part, water is going through a special protein called an aquaporin, and anything that's large is having to go through a protein or come in through endocytosis. So, in a nutshell, what can cross the membrane? Small and nonpolar. And why? Because of these nonpolar tails. They are very, very picky. Like I said, the heads will kind of look the other way sometimes, the tails will not. They will not let anything through that they don't want through. All right, I can compare and contrast passive and active transport. Okay, they are both types of transport, obviously, because it's in the name. And so they're both ways that things can get into and out of the cell. Okay, they both deal with concentrations, concentration gradients. Passive are usually going to destroy your concentration gradient. Active are going to create or reinforce that concentration gradient. So passive, you're always going to be moving from high to low concentration. Okay, whereas active, you're going low to high concentration. So over here, this would be my high, this would be my low. Let me write the whole word. So passive, you would see, we'll do passive in pink. Passive, you would see the molecules moving this way because they are trying to escape that crowded scenario. And so they're going to try to move over to the lower concentration until it's equal. So they're going to keep moving until you have equal amounts on each side, and then they won't move, or they'll move back and forth evenly. Active concentration is going to push against that. They're going to create a concentration gradient, or if there's already one there, they're going to reinforce it by adding more of those molecules into that high concentration. This is not what they want. These guys are already crowded and they're saying, no, we don't want any more. And active says, I don't care, take some more. And so it's going to have to use energy to do that. So energy is required. And like I mentioned earlier, it will reinforce that concentration gradient. It'll make it more different. So even higher here and lower here because you're pumping more that way. Passive, on the other hand, does not require energy, because it's just falling through. It's just due to the random movement of molecules in the direction that they want to go anyway. And so it will usually destroy that concentration gradient. Okay, because a concentration gradient by definition means you have different concentrations on each side, but passive is going to push these over until it's equal. And so when they're equal, there's no more gradient. Okay, and so those are some of the similarities and differences between active and passive transport, as well as why certain things can cross the membrane and other things cannot. And I hope that helps. If you have any more questions, let me know.